Welcome back to this course on organic chemistry and biology and drug design. So far we have covered various aspects of a very important biomolecule like the proteins and their functions, and we have concentrated mainly on their catalytic activity, that are the enzymes. Okay. So, we covered the structure of proteins, various hierarchical structures, then how to determine the sequence and how to separate them, and finally, the enzymes and the kinetics and their inhibition profile. Then we went to one aspect of synthetic biology, and we have covered the catalytic antibodies or enzymes and how to design them and how to catalyze different organic reactions. We have just given few examples. Okay. Now, today we will now switch the gear and change to another topic, another very important topic, that is the nucleic acids. Okay. The nucleic acids, you are aware of that what are these compounds. They are consisting of sugar, phosphate and a base, and then this is the monomeric unit which then joins together one by one and finally give the what is called the nucleic acids. Okay. The nucleic acids has a history behind it, that how was it discovered. In 1869, Frederick Meissner, he isolated what he called nuclein from the nuclei of some particular cells, that is pus cells. Pus cells means where we have infections, the cells which are excreted are called the pus cells. So, these pus cells, from these cells he isolated a product which he called nuclein, and later on nuclein was shown to have acidic properties, and hence it was called nucleic acid. So, the term changed when it was found that it has got acidic, it is basically an acid. So, acidic properties, and that is why this is called nucleic acids. Now, two types of nucleic acids we know are there, and one is deoxyribonucleic acid, that is the DNA, that is usually the compound which dictates or which stores the information of making the required molecules to perform certain functions. Okay. So, that is the deoxyribonucleic acid, or to specifically deoxyribonucleic acid stores the information that via the ribonucleic acids, then it transmits the information and make the proteins that are required to maintain the living system. Okay. Now, DNA is found, you know that there are again broadly there are eukaryotic cells, that is the higher organisms, and there are prokaryotic cells, that is like the bacteria. Okay. So, DNA is generally mostly found in eukaryotic cell in the nucleus, that is there is a separate zone which has got its own cover, and within that the DNA is found with small amounts, that is written here, small amounts in mitochondria and chloroplasts. Okay. But mainly in the nucleus for the eukaryotic cells. For prokaryotic, RNA on the other hand, RNA is found throughout the cell. So, throughout the cytosol RNA can be found. So, that is one difference between the DNA and RNA. DNA is very localized, whereas RNA is not. In case of what is not written here, that in case of your prokaryotic cells, then you do not have any particular nucleus. So, the cell has no inner compartment, it is not compartmentalized. So, DNA, RNA and protein are all together in the cytosol. Okay. So, that is the difference, because the lower, that is the lower level of organisms. Okay. Now, the nucleic acid, as I told you, there are two types, okay, but they are monomeric, but they are polymers. They are polymers of what? They are polymers of what are called polynucleotides. That means their monomer is the nucleotide, and the nucleotides are joined together, that is their building blocks. The building blocks are the nucleotides, and they are joined one after another and then form what are called polynucleotides, and that is what is the nucleic acids. Okay. Now, what is the question is, what is the nucleotide? The nucleotide is nothing but it contains a sugar, a base and a phosphate. So, you have a base here, sorry, you have a base here, and you have a sugar, a particular type of sugar, and a phosphate linkage. Okay. So, that what makes a nucleotide. So, nucleotide I can write that you have a sugar, you have a base and you have a phosphate. So, this is a nucleotide. Okay. Now, the question is, what are these bases in the nucleotides? What is the sugar in the nucleotide? And that is also written here, that the sugar is ribose, that is a 5 carbon monosaccharide. Okay, that means aldopentose is a ribose. Okay, or deoxyribose, that means a ribose which is stripped of one hydroxy group from some position. Okay. So, the sugar is either ribose or deoxyribose, and then the bases are basically virtually 4 types, but classification wise they are classified as purine bases or pyrimidine bases, depending on the type of heterocycle. This purine and pyrimidine are the names of the heterocyclic system that are present in the bases. So, what is your purine? Purine is nothing but, this is your, this is called pyrimidine, and purine, when you add a an azole, then it becomes purine. So, this is your purine, and pyrimidine already, purine is nothing but azole fused 1,3 azole fused pyrimidine, and the other one is only pyrimidine. So, there is no, so this is the skeleton, this is called pyrimidine. So, these are the bases, two of the bases will have this heterocyclic ring, and the other 2 bases have this heterocyclic ring. Now, you will notice there are actually, rather than 4 bases, there are 5 bases, and that is because there is a difference in one of the bases in RNA as compared to the DNA. That means, in deoxynucleotide, the bases are adenine and guanine, and the pyrimidine bases are cytosine and thymine. They are written as AGCT, whereas in RNA, instead of thymine you have uracil. So, RNA has the bases A, G and C, U, whereas DNA has bases A, G, C, T. Okay. Now, that is the so, I will show the structure of the bases, that is very important. Interestingly, there are only 4 bases in a particular type of nucleic acid. Okay. And these 4 bases, their arrangement, their sequence actually dictates how the information is stored in the DNA, or how the information is stored, that is dependent on the sequence of these bases. It's basically, if you look at the your electronic system, the computerized system, what you have is a binary numbers 1, 0 and 1. But if you arrange, you can arrange this 0 and 1 in different possible ways, and that gives different types of signals. Okay. So, it is the same thing, you arrange all these bases in different types of ways, and that gives different types of information stored on the DNA. Okay. So, the nature did not have many of these bases, did not use many bases, only 4 bases to store the information. Okay. Now, what about the sugar? The sugar is basically same, ribose in case of RNA and deoxyribose in case of DNA. So, this information that is stored, that depends on the sequence of the bases, because the sugar is constant. In DNA it is deoxy sugar, in RNA it is the normal sugar. Okay. So, the sugar part is same in all the DNA, the phosphate part is same. So, what changes is the base. So, this sequence of bases, when they are connected, these polynucleotides, that decides what information is going to be stored. Okay. Now, let us see what are the structures of these bases, or what is the structure of the sugar. The sugar, as I said, is a ribose. So, that will be a pentose, specifically an aldopentose. So, that will have 5 carbons, and it is present in the furanose form, remember, because there are 2 forms that are possible, furanose form. So, 5 membered ring form, and that forms a hemiacetal. Okay. Actually, pentose, we should, if you write the wedges, so you have the wedge here, you have a wedge here, you have a wedge here. I am not writing any stereochemistry, wedge here, and there is a wedge there. So, that is here that is aldopentose with a particular configuration that becomes ribose. Okay. So, that is present in DNA, that is present in the furanose form, that means the 5 membered ring. So, now if you put the hydroxy in the proper perspective, that means with proper stereochemistry, what you see that this is ribose, where this is what is called the anomeric carbon. Okay. This is the anomeric carbon through which you make the what are called glycosides. So, if you replace the wedge and place something here, a carbon, that is called a C glycoside. If you put a nitrogen here, that will be a N glycoside, O glycoside. So, all these can be possible, but that are all through this anomeric carbon. And on the other hand, also check that this hydroxy is alpha. Alpha means it is below the plane of the ring, and this is also alpha, and this CH2H is beta. So, that makes it ribose. If you change the stereochemistry, that is no longer ribose. So, nature uses this sugar to make the backbone of the nucleic acids, and deoxyribose is the one which will lack one hydroxy group, and it is found that this 2 hydroxy group, that is actually, if you name the ribose separately, then this will be your 1, that will be your 2, that will be your 3, 4 and 5. So, it is the 2 hydroxy that is gone. So, this is 2 deoxy ribose, because there are many deoxyriboses, but the ribose that is used to make the backbone of the DNA is 2 deoxyribose. Okay. So, that is all about ribose. But these numbering system, one thing I should mention here, because when you attach the base, and the base has lot of carbons and nitrogens. So, the question is whether this same numbering system will be retained or it will change, and there is a slight change, because base takes the numbering as 1, 2, 3, 4, and the sugar takes a numbering 1 prime, 2 prime, 3 prime. So, this will become prime numbers 1 prime, 2 prime, 3 prime, 4 prime. Okay. So, let us now go to the, so now we know that there is a sugar moiety, and that sugar moiety, this is the that C5OH, which will ultimately become 5 prime when the base is attached. So, that has a phosphate, and a phosphate linkage connects these sugars one after another. You see, this is the connection when it makes the nucleic acid backbone. And if you inspect it even a little more carefully, then you will see that, maybe we can go to the next one. This says that the phosphate group joins the third carbon. See, what is the joining? This is the third carbon of the sugar, and this is the number 5 carbon of the sugar. So, when the connection is made, when these sugar units are joined via phosphate, so you see the 3 prime hydroxy of one sugar unit is joining with the 5 prime hydroxy via a phosphate linkage. We will also check what is this phosphate linkage. We have just said in a block like P, but there has to be oxygen on the phosphorus. So, we will do that. But anyway, remember, it is the 3 prime 5 prime, that connection, or 5 prime 3 prime. You can also say 5 prime hydroxy connected to 3 prime hydroxy via the phosphate, or 3 prime going to the 5 prime. So, that is the important thing. Third carbon of one sugar is connected to the 5th carbon of the next in line. Now, you add to the base to make it a complete nucleic acid. Okay. So, now the bases are added. Where the bases added? The bases are added at the anomeric carbon, that is the C1 carbon, the first carbon of the, the first carbon, this is the C1 carbon, and they are all connected, all the bases are connected one after another, but all are connected at the C1 carbon. Another point to note is that whenever a sugar is there, the anomeric weight can take a beta orientation, that is the beta glycoside we call, or it can take an alpha position also. So, that is the alpha glycoside, but in this case remember these G are actually, it should be written like this, they are all connected like this, G, this is C, this is C. So, they are all beta glycoside, but these are N glycosides, because this is not connected to the carbon atom, this is connected to the nitrogen. I told you glycosides are classified according to the atom that which is connected to the anomeric carbon. So, if it is carbon, that is carbon C glycoside, if it is oxygen, that is O glycoside, and if it is nitrogen, that is N glycoside. Okay. Now, so this is the arrangement, how these nucleic acids are arranged when they make the polymer. Okay. Now, this you know that Watson and Crick, when the DNA was isolated and people tried to know what is the actual 3-dimensional structure of the DNA, then there were a lot of efforts that went parallelly. It was one group at London, and then there are other groups at Cambridge University. Watson and Crick was working there, and then there are other groups at California where Linus Pauling was trying to make the structure of the DNA. So, there was a hectic competition between all these groups. Now, to know the structure of these molecules, what we need 3-dimensional structure is a X-ray diffraction diagram. What is about pattern? That means you have to isolate pure DNA. Actually, DNA is isolated not like crystals, but it is like a fiber, and when the fiber, you can take the X-ray data of the fiber, and from that X-ray data, it is not easy, but it was solved ultimately, and it was Watson and Crick who announced in a landmark paper that in 1953 that the DNA has a helical structure, and not only helical structure, there are two helical strands which are joined together by weak forces, and also they at that time they said that it is the guanine base, that is one of the purine base, which is hydrogen bonded. These lines are basically hydrogen bonded to a pyrimidine base, but specifically G forms hydrogen bond with C and A forms hydrogen bond with T. So, what happens that when one strand has all these sequences G, C, A, T, T, then the opposite strand, that is called the complementary strand, that will have bases which can form hydrogen bond with the base that is present in the original primary strand. So, now the sequence of the complementary strand you can write, because there is T and T recognizes A and forms two hydrogen bonds interestingly, and then T A, then it as it is A, that is T, then C G, C G and G C. So, that way the two strands are attached. Now, the force of attachment, that means what is the force by which these two strands of DNA are attached to form a double helix, are not only the hydrogen bonds that attaches the two strands, but also something what is called that these rings are primarily aromatic in nature, and these when the aromatic rings lie one after another in a parallel mode, then there is some stabilizing interaction which is called pi stacking interaction. So, this pi stacking, because all the bases are like in a parallel mode and parallel to each other, and then they form what is called pi stacking. So, this pi stacking also stabilizes the DNA. Now, this when there are two strands in the DNA, that is called double helix, that we know, but sometimes we call that as a duplex DNA. Okay. So, we know that the duplex DNA is formed because of this hydrogen bond, and nature has devised this mechanism because that actually ensures that these bases are all inside, because these are the storehouse of our information. So, what happens, these phosphates are actually exposed outside, but the bases are stacked together inside, inward. So, that is basically protecting the DNA from outside agents to damage these structures. Okay. Now, the next question is, this we have already said, A pairs with T, G always pairs with C. So, that means a purine pairs with a pyrimidine, and the most important point here is the complementarity, and there are two important terms, and the anti-parallel nature. Okay. When you join, let us see, there is such a, when you join a sugar unit, now let us clarify what is this this phosphate, how they are linked together. This is oxygen, and then you have a phosphorus that is connected to double bond O and O minus, and then this is the oxygen from the 5 prime position, and this is the base, and then again another one comes like this. So, you have the phosphate and O, this is your sugar unit. So, it goes like that, that is another base. Okay. Now, there are few things to notice. One is that you are starting the DNA at the, because we are writing it in from top to bottom, the strand is extending like this. So, at the top you have here the 5 prime which will be free at, because when you have the DNA strand, so on one side your 5 prime which is free, and on the other side will be your 3 prime wedge, that will be free because the length has to terminate at some point of time. The DNA has a defined length. So, at one end you have a 5 prime end and the other end is called the 3 prime end. What is 5 prime? Like in amino acid peptide, what you have a N terminus and C terminus. Okay, those are free. So, here which hydroxys are free? On one end the 5 prime hydroxy is free and the other end the 3 prime hydroxy is free. So, we say that this runs from the 5 prime to 3 prime direction. So, whenever we write a piece of DNA, we have to mention, for protein it is usually understood that this is the N terminus, that is the C terminus, but in DNA what happens, usually it's better that you mention. There is no rule that the left side is always 5 prime and the right side is 3 prime. It could be the other way around. Okay. So, when they form the double helix, when the 2 strands are joined together, then first of all I showed that the force between this attachment of the 2 things are all weak forces that all the G's are paired up with the C via 3 hydrogen bonds. I will show you where are these 3 hydrogen bonds, and A attaches with T. Although each hydrogen bond is weak, but the sheer number of hydrogen bonds, the number of weak interactions is so enormous that DNA becomes very stable. But also remember that there is another stability factor which is called the pi stacking interaction between these base pairs. Okay. Between the underlying base pairs you have a pi stacking interaction. Okay. Now, so basically there are 2 strands. Whenever I talk about a DNA, a double helix DNA, there are 2 strands and there will be a hydrogen bond in between. Okay. Different bases, complementary bases are there and there are hydrogen bonds between. What is antiparallel? Antiparallel is that one strand runs from 5 prime to 3 prime in one direction and the other strand runs from the, from the same direction, if you follow 3 prime from left to right, this is 5 prime to 3 prime, this is 3 prime to 5 prime. That means they are running antiparallel. This is what is called antiparallel. So, first of all, the bases here are complementary, and the second one is the 2 strands are running in opposite direction. One is running from 5 to 3 prime here and the other is the from the opposite direction. Okay. Let us see now where are these. Now, this when before the structure of DNA was known, there was another scientist whose name is Chargaff. Chargaff, he actually was studying, he took a piece of DNA from different sources and he was studying the percentage of A, that is adenine, percentage of guanine, percentage of cytosine and percentage of thymine. These are the 4 bases. So, he was measuring the percentages of this. Now, this turned out to be a very important experiment. What he found, that if you take suppose from man, he isolated some DNA, the percentage of A is 30.9 and percentage of T is 29.4. So, that is within all experimental error they are same, there is not much different, and the percentage of G is this and percentage of C is this. So, G is equal to C and A is equal to T. Okay. So, you take another, say some other species, sheep, 29.3 is A and 28.3 is T, and these are two are almost same, 21.4 and 21.0. So, it was after studying the DNA of various species, he came to the conclusion that it is the A by T will be 1, it is all nearly 1, and G by C will also be nearly 1. That means, A percentage of A is equal to percentage of T, percentage of G is equal to percentage of C, and that means so A by T is 1 as I said, and A by and G by C is also 1. you can also find out what will be the ratio of A plus G and C plus T. Now, A is basically the percentage of A is equal to percentage of T. So, if that is x, so that will be also x. Percentage of G is suppose y, so percentage of C is also y. So, that means it is 1. So, A plus G, not only A by T is equal to 1, G by C equal to 1, it is also A plus G divided by C plus T is also equals to 1. Okay. Now, this is the famous X-ray crystallographic picture that was taken by the London group. I was telling that there are 3 groups at that time were heavily competing with each other. So, there was one lady scientist, his name is Franklin, Rosalind Franklin. Franklin in 1952 took this is the X-ray picture. Okay. That is the best X-ray picture that one could get at that time. So, Franklin got this picture, and Maurice Wilkins was also working along with him. He was a colleague of her, and he showed this picture to Maurice Wilkins. And Wilkins, this is the what is the truth behind it, nobody knows, but somehow Watson and Crick got hold of this picture, Franklin's picture photograph, and then because Crick was extremely Crick was very good in X-ray crystallography. So, he immediately from showing this, after seeing all these, and he knew that the DNA is helical. Only problem was they could not come out with an explanation that why the percentage of A is equal to percentage of T. The Chargaff's rule, that percentage of A equal to percentage of T and percentage of G is equal to percentage of C. So, that they could not come out. Okay. So, ultimately with help of with both of them worked together, and there is a famous picture I will show you between when they, yes, these are the 2 gentlemen. This is Francis, this is Crick, and this is Watson. So, they actually virtually made the DNA that with building blocks, the models, like I have the models also. So, but with it requires a numerous number of atoms, and they connected it and ultimately found that it is a kind of the helical structure. That means the twisted stairs that you see in the fire exit. So, some kind of that. So, they one night they worked whole night and finally came out with a conclusion and or the structure of the DNA, and they at that time they proposed that G is pairing with C and A pairing with T, and that solved, because it is a double helix, that solved the that will immediately tell you that the percentage of A will be percentage of T and percentage of G will be percentage of C. So, they announced all this in the next morning, that was 28th July, and then 28th February, sorry, which is also a very important day, the National Science Day, when C.V. Raman also discovered his Raman scattering. So, a very famous day, but they did not publish at that time, they actually announced it on 28th of February, that they solved the mystery of the life, because they immediately realized that this discovery of DNA structure will lead to the subsequent happenings in a living system, or how the living system functions, how the memory is stored, how from one generation message is passed to another generation. So, everything. So, this is what is called one of the landmark discovery in biology, and the whole molecular biology area started because this is, you are seeing the molecule, you are seeing the system at the molecular level. Okay. So, that is the that is what I just did not show you, is the structure of these bases, because this you should remember, all these Ultimately, it is 5 bases. Uracil is present in RNA, and instead of uracil, thymine is present in DNA, and these are the structures. So, this is the purine nucleus, and then you have an amine. Remember the numbering system, that is also very important. The numbering system, it starts from here 1, 2, 3, 4, 5, 6, 7, 8, 9. So, this is 6 amino purine, that is called adenine. So, here it is 1, 2, 3, 4, 5, 6. So, 6 keto, 6 oxo basically, that is and then 2 amino purine is what is called guanine. Then, so that is the purine bases, and you have cytosine and thymine and uracil, they are all pyrimidine bases. That means, a type of benzene ring where the nitrogens are in a 1-3 relationship. So, this is cytosine. Again the number, it starts from here 1, 2, 3, 4, 5, 6. Now, sometimes it is really difficult, but you should remember this numbering, because all the time when some reaction takes place on the bases, we generally go by just we will say that the C3 works, C3 has reacted, or N7 nitrogen, N7 is protonated. So, you have to remember this numbering. So, there is this, but in pyrimidine always this is 1, and then it goes to the other nitrogen 1, 2, 3, 4, 5, 6. So, that is same in every one, cytosine and uracil. So, only thing you have to know is that which nitrogen is taken as 1. In pyrimidine this is the one, and in purine this is the number 1 carbon, and then you go to this other nitrogen 1, 2, 3, 4, 5, 6, 7, 8, 9. So, that is that numbering is important, and the Watson-Crick base pairing, that why this is these are forming hydrogen bonds, because exactly in hydrogen bonds what you need is a donor. Donor means some electronegative element with a hydrogen, and an acceptor. The acceptor is another electronegative element. Now, if you look at the structure of thymine, this is the structure of thymine. Thymine has a methyl, this is carbonyl, and this is a pyrimidine ring. So, it has got a NH, it has got a carbonyl and a carbonyl, and adenine, actually this is there is a double bond here. Adenine, there is this NH2. Okay. So, now this NH is a donor, and thymine carbonyl is well placed to make a hydrogen bond with this NH. And on the other hand, this is NH on the thymine, and that NH is becomes a donor. So, this is acceptor, that is donor, and that is now well placed to make. Well placed means the distance criteria, that these are all less than 3 angstrom. So, that they form the hydrogen bond. So, this nitrogen forms the hydrogen bond with the NH, and this carbonyl forms the hydrogen bond with the NH2 of adenine. But the other carbonyl, that is an acceptor, but unfortunately there is no other group which can donate hydrogen to this carbonyl. So, this has only 2 hydrogen bonds. On the other hand, if you take the GC pair, so if it is, say G has, if you look at adenine, adenine has one donor atom NH and an acceptor nitrogen, but here nothing is there. That is why no hydrogen bond is formed here. But in guanine you have this is acceptor, this is donor, and this is donor. And if you look at cytosine now, if you look at cytosine, cytosine structure is this. So, this is your donor, that means your acceptor donor, that combination you need. This is your acceptor. So, again donor acceptor combination is there, but unlike adenine, in guanine you have another functionality here, which is another NH2, which is a donor. As I said, there is nothing here. So, you do not have any hydrogen bond here, no hydrogen bond, but here you have a hydrogen bond because there is a NH2. So, that is why there are 3 hydrogen bonds formed in the between G and C. So, basically they are so complementary to each other that they fit electronically, geometrically, and then form the hydrogen bonds, and that is the basis of the basis of formation of the double helix. Okay. Now, these 3 hydrogen bonds versus 2 hydrogen bonds, that will be important, and in the next session I will tell the importance of this, the GC and the AT and their percentages. Okay. Thank you.